Well, respected moderator, although she is absent, distinguished guests, colleagues, attendees, ladies and gentlemen, at the outset, I would like to thanks to the organizers for giving me a chance to be a speaker over here. I'm thankful to the chairperson of my institute, Madam Amina Al-Khaidan, for encouraging me to present this topic. Well, the topic, as you know, is herbal medicine quality control studies. Well, plants have always been a common source of medicament, either in the form of traditional preparations or pure active principles. In a survey done by World Health Organization, it has been estimated that 80% of more than 7,000 million, that is 7 billion inhabitants of the world, rely mainly on the traditional medicine for their primary healthcare needs. And it can safely be presumed that a major part of traditional therapy involves the use of plant extracts or their active principles. Well, before proceeding, I requested before that the lecture should not be recorded because most of its studies are under publication. Then, an immediate response in absence of moderator. Is it being recorded or not? I requested it should not be recorded because most of its matter are under publication. Okay, I'm continuing. Well, as I said before, that 80% of more than 7,000 million or 7 billion inhabitants of the world rely mainly on traditional medicine for their primary healthcare needs, and it can safely be presumed that a major part of traditional therapy involves the use of plant extracts or their active principles. Well, in developed countries too, plant-derived drugs are considered important. For example, in the United States of America, 25% of all the prescriptions dispensed from community pharmacies contain plant extracts or their active principles. Well, the World Health Organization has reported that more than 120 plant-derived drugs are being used in the modern medicine, and the major pharmaceutical companies are now performing research on the plant parts for their potential medicinal values. According to the global industry analyst, if we see the growing importance of the herbal medicines and the plant sector, it is, if you see it from 2000, you see the figures, 43,000 million world market sales at that time. And now in 2022, it was $125 billion, whereas it is expected that it will be $196 billion by 2025, and 5 trillion by 2050. Of the conservatively estimated 300,000 to 400,000 species of the fire plants that exist on Earth, almost 10 to 20%, that is 35,000 to 70,000, have been employed in traditional medicine. Well, the efficacy of these drugs is due to the presence of one or more biologically active chemical constituents. But fortunately, quite a number of these drugs are frequently adulterated with cheap, less potent, and spurious plant materials, either posing danger to the public health or they are not so efficacious as expected. Because the therapeutic efficacy of these drugs will depend mostly on the purity and economic availability. For the next slide, it's the movement is not there. What is the matter? Well, because the therapeutic efficacy of these medicinal plants will depend mostly on the purity and economic availability. Well, in the recent year, with ever-growing commercialization in the field of herbal medicines, there has been an instant demand for the quality control of the drugs used in the system. Sorry. For this, standardization is usually recommended as a solution to the problem. The studies on the identity, purity, and quality of the genuine drugs will enhance information in checking the adulteration. A set of standards no doubt be deterrent on substitution and adulteration. Rather, it will be an aid to the drug law enforcement. But what is conveniently overlooked is that the standardization of the materia medica of the medicinal plant itself is a difficult task. And therefore, before dealing with this topic, let us recapitulate what is meant by standardization. Standardization may be defined as the elimination of unnecessary variety. In other words, it implies the collection of the relevant data, putting it down in a logical sequence, eliminating the varieties and inconsistencies before formulating the standards. So I was telling that standardization may be defined as the elimination of unnecessary variety. In other words, it implies the collection of relevant data, putting it down in a logical sequence, eliminating the varieties and inconsistencies before formulating the standards. Well, in order to appreciate the problem facing standardization of herbal medicine, one has to examine the situation in totality. Well, in the present slide, you can see one has to examine the situation in totality. The term totality stands for examining the problem of standardization from the time of plantation of the drug to the time it is either dispensed to the patient in crude form or in the finished product. Well, the word standardization includes various problems and fields of study. The standardization includes a study from birth of the plant to its clinical application. The vast study and involves various disciplines. Obviously, the scientists are looking after isolated parameters, but one should have a detailed idea of the combined approach. The study of the plant includes its cultivation, source and period of collection, identification, storage, chemical composition, pharmacological study to its clinical application. Agricultural practices. Well, selection of medicinal plant. I'm not going into detail, although each topic it needs elaboration. But since the time has given to me 20 minutes only, so I'm only reading selection of medicinal plants, documentation, seeds and propagation material, cultivation. And these are very important topics, as you know, that, for example, in documentation, in addition to keeping documentation on the botanical identity of all medicinal plants and aromatic under cultivation, the growers and producers shall prepare botanical voucher specimens for submission to the regional or national herbaria for authentication purpose. Let us come to the cultivation. This is a very important point, as we know that different plants have different habitat. The habitat of the herb will ensure the uniformity of activity in relation to the source. The very first step should therefore be the cultivation of the medicinal plants under controlled conditions. The prerequisite for this is the collection of authentic specimens of such plants as are used in the subsequent investigation of the methods for large-scale cultivation. Well, cultivation of medicinal plants requires intensive care and management. Site selection. Medicinal plant materials derived from the same species can show significant difference in the quality when cultivated at disparate sites due to the influence of soil, climate, and other factors. These quality differences may be physical in appearance or may be manifested by the variations in the pharmacologically active constituents. Ecological environment. The cultivation of medicinal plant may affect the ecological balance of flora and fauna of the environment and vice versa. I'm cutting short. The soil should contain a sufficient amount of nitrogen, phosphorus, minerals, organic matter, or other elements. Human excreta should never be used. Animal manure should thoroughly be checked, and permitted agrochemicals should only be used. Irrigation and drainage should be controlled and carried out in accordance with the need of the individual medicinal plant species in its various stages of the growth. Plant maintenance. Any chemical used in the growth and protection of the medicinal plant should be kept to a minimum and should be applied only when no alternative measures are available. Measures as stopping, budgeting, pruning, and shading may be used to control the growth and development of the plants. Pesticides and herbicides application should be avoided as much as possible. Harvest. Medicinal plants should be harvested during the proper season or time period to ensure the production of the quality of the source plant material and the subsequent finished product. The best time for the harvest should be determined according to the quality and quantity of the biologically active constituent rather than the total vegetative yield of the crop. Personnel. The producers and the growers, that is the farmers, they must have a knowledge of the medicinal plant they are growing, that is the botanical identification, cultivation, characteristics, soil type, pH, etc. High degree of personal hygiene is required. Field collection practices. Prior to the field collection, the population density and geographic distribution of the target medicinal plant must be identified. Topographical map of the collection site should be prepared. Field collection team and their responsibilities should be clearly chalked out. Permission and collection permits. That is, the prior permission for the collection should be obtained. Selection of medicinal plant. That is, collected medicinal plants should be the same as those described in the National Pharmacopoeia. Collecting technique. Ecologically non-destructive means of collection should be employed. Good agricultural and good field collection practices, they are ethical and legal issues like intellectual property rights, threatened and endangered species. Well, the proper storage and preservation method of the plant material are essential for maintaining a high degree of the quality of the drug. Some plants are particularly liable to deterioration if they become damp during shipment or storage. There is an example of cascara leaf. Yes, it is there. For example, cascara leaf. Medicinal plant is not well packed. They absorb moisture, which triggers enzymatic activity, leading to the spoilage and storage. As aware, the organic constituents of the plants have a limited shelf life. These compounds undergo degradation under the influence of heat, light, and moisture, particularly when standard conditions for the drying storage are not maintained. Well, after all, identification of a plant is most important because nowadays the medicinal and non-medicinal plants, the adaptation is a common practice. So closely related medicinal and non-medicinal plants should be clearly distinguished on the basis of their minute differences. Well, misidentification of the two Chinese plants, as you can see in the figure. The slimming preparation is believed to be responsible for serious toxicity, that is renal intestinal fibrosis, experienced by some 70 Belgian women. 30 of them needed dialysis or kidney transplantation. And this was a confusion between Stephania tetrandra and Strelachia fangchai and was thought to have been arisen due to the similarities between these two plants. As aware, Strelachic acid are well known for its nephrotoxicity in human and several animal species. They are also potent carcinogens in the rodents, and that is why restrictive regulation was put forward in the year 1981 in Germany for the preparations that contains strelachic acid. Coconut root. Sometimes identity by ordinary taxonomic means is a problem and requires various other fields of study. Let's take the example of a plant which is very controversial. The botanical identity of this medicinal plant, which is sold in the crude drug market in many countries in the form of isolated underground part with some remains of radical leaves, is highly controversial because some 15 species belonging to 10 genera and three families, they are all being sold by the same trade name. It is known as Ratan Jot in India and Pakistan, Yadlang in Afghanistan, and Abu Khalsa or Saleh Imam in Arab countries. These all are equivalent vernacular names. The pharmacognostic and chemotaxonomic studies of the different boryginaceous species from, you can see from 3 to 7, the number 6, or maybe a nobilis is the actual required genuine plant material. Organized drug. Pharmacognosy remains best means for the identification of single drugs, especially those of the plant origin. And the drug used as a whole plant or any part, it's easy to identify the drug botanically. But for an unorganized drug, or when the drug is reduced to powder form, greater degree of exercise is needed on the part of the pharmacognosis. Moreover, pharmacognosy by itself doesn't provide all the information. And identification does not stop with the standardization, doesn't stop with the identification, but extends to quality and potency. Recourse to the physical and chemical method of standardization becomes necessary rather a must. Well, chemical standardization is possible if we know the limits of variability of any characteristic constituent of the drug, but present in a measurable quantity. The constituents present in small amounts or too small amounts may not prove useful for standardization purpose. The isolation of a single specific compound is itself a difficult task, as you know. To press them into service for standardization purpose is even more challenging. The standardization of herbal drugs on the basis of chemical studies has got its own advantages. For example, exactness of results, quick and less time-consuming observations, and recovery of the material where the drug to be identified is in small amounts. There can be several methods for the standardization of herbal drugs chemically. However, it depends upon the nature of the drug under study as to what particular method should be applied. Although no single method can be suggested for standardization, but there are certain common parameters which can be established. This is a general format for the standardization of the herbal drugs. Well, preliminary examination. In the case of the whole drugs, the macroscopic and the sensory characters are usually sufficient to enable the drug to be identified. The general appearance of the sample will often indicate whether it's likely to comply with such standards as percentage of seeds in colocynth above, ash in valerian, and alcohol insoluble matter in asafoetida. Well, the price of certain drugs will depend mostly on the size and color, which are not necessarily related to the therapeutic value. And this applies to such important drugs as on the slide you can see chamomile, ginger, senna, rhubarb, and so on and so forth. Now, some specific organic and inorganic constituents have to be identified. The identity, purity, strength, stability, and assay have to be carried out. As we read here, foreign organic matter, pesticide residue, radioactive contamination, and determination of heavy metals. There are important protocols for the identity. These are the important protocols for the identity of a plant. Microbial contamination. Aerobic bacteria and fungi are normally present in the plant material, and they may increase during the course of harvesting, storage, and processing. The different limits are set, as you can see in the on this slide. Different limits are set according to the use of the material and the material itself, and therefore the toxic metabolites may be determined after using a suitable cleanup procedure. No drug may be assayed for a particular group of constituents. For example, the total alkaloid in belladonna. Changing the slide. For example, total alkaloid in belladonna, total glycosides in digitalis. An alternative, it may be necessary to evaluate the reserpine content in which is distinct from the total alkaloidal content of Rolfia serpentine. Chemical constants have to be identified. Successive extraction has to be performed in increasing order of polarity. For example, petroleum ether, benzene, chloroform, ethyl acetate, acetone, methanol, and water. Figure 20. By the use of the chromatographic technique, it's very easy to find out the number of the constituents present in various extracts of a drug. The retention time and the RF value of a compound determined under specified conditions is characteristic, and they can be used as an identity. The molecular weight, below slide, the molecular weight and the fragmentation pattern will further facilitate the identification. Fingerprinting of Rolfia serpentina, very famous plant. All of you must be the marker compounds. Actually, marker compounds are the compound of interest for control purposes. Although it's not easy to find out them, you can see a long procedure. Laboratory experiments are being carried out to find them, but they are basically compounds of interest for the compound of compound of interest for control purposes only, irrespective of the fact whether they have a therapeutic activity or not. Now, if we see the inter and intra-species variations in Ami Visnaga and Ami Mages, two examples I have taken. The transverse section of the mericarp of Ami Visnaga, Ami Visnaga and Ami Mages, when the fruits of Ami Mages are occurred as admixed and substituted with the Visnaga, they can be distinguished by the secondary ridges and by the absence of lacunae outside the vascular strands as seen in the transverse section. They can also be identified through the thin layer chromatographic studies. Ami Mages, you can see the compounds present in the Ami Mages, and also Ami Mages contains 5 and 8 methoxysoralin, whereas Ami Visnaga contains stellene and visnagine. And this can be interpreted through thin layer chromatography, high performance liquid chromatography, and by the co-chromatography also. These are the different, for the extraction of these plant material, these methods are being used. Here a number of the samples can be tested. If almost some 100 samples, if in a small amount have to be extracted, we can use this procedure. This soft fillet extractor, as you know, soft witchy rota vapor, supercritical fluid extraction method. So basically, these extracts can be validated through these critical process parameters. And you can see by the side that the freeze dryer is also there. Separation and isolation of chemical compounds, we use the planar chromatography for the analysis of medicinal herbs. Sample application is done by the Saga machine. Chromatograms are being developed in same. Evaluation is done through these methods. Evaluation also, the documentation is done, and then we are using all these machines through. You must be knowing all these machines. The UV, different UV spectrophotometers, and IR, UV, NIR, and I'm not going to go in more detail, GCMS and MS, and all these instruments. So by these, we find out the identification of any plant. For example, garlic, it can be differentiated through its fingerprinting to the onion, because the chemical constituents, as you see in the fingerprinting, it's quite different. Current good manufacturing practice is a system for ensuring that the products are consistently produced and controlled according to the quality standard. It is designed to minimize the risks involved in any phytopharmaceutical production, that is the herbal production, that cannot be eliminated through testing the final product. Composite herbal formulations have different dosage forms like pills, semi-solid, ointment, oil, liquid, you know all these. The very important thing is that in order to ensure the quality of a licensed herbal medicine, it is essential, therefore, not only to establish the botanical identity of the herbal ingredient, but also to ensure batch to batch reproducibility. Right, there are three stages of standardization: single herbal drugs for method of processing and standardization of the finished herbal products, as you know. Now, the most important thing is coming as main, actually, what are the main and important objective of herbal drug standardization? To evaluate the standards of single and composite herbal formulation, which would act as a guidelines for the manufacture of herbal medicines. The standardization of herbal drugs will further facilitate prosecution of offenders. It helps also in official compendium of indigenous herbal medicines and publish official monographs. Well, now the important thing is, unfortunately, many of these licensed herbal products have been found to contain non-prescription and even prescription drugs. Why not forwarding immediately? Okay. Yes, you can see here practically in thin layer chromatography, many samples of the herbal drugs were taken. Number 3 is the standard sildenafil citrate, and you can see that the concentration of all the sildenafil citrate, that is Viagra, is more than the standard in all the sample except that of number 2. And another UV light, you can observe that the other phytoconstituents are very, very meager as compared to the sildenafil citrate. There are many constants of the quality control of the herbal drugs. As you can see, it depends on the wildlife, regulatory aspects are involved, herbal pharmacopoeia not available, adulteration and substitution, no certification of raw material, lack of trained manpower, lack of safety evaluation studies, poor investment in research and development. So, although the standardization, today the standard might become obsolete tomorrow, although quite a lot of work has already been carried out in this respect globally, but still there are certain dark recesses which have to be eliminated using high throughput drug technology like random amplified polymorphic DNA fingerprinting, hyphenated NMR studies. So these basically, if we are using, and people are using it nowadays, DNA micro application of natural products for drug discovery and development. So basically, let me say before that these genomics and metabolomics seems to be the future of herbal drug industry or medicinal plant industry, irrespective of the fact that these are very costly, their prices, and all manufacturers cannot afford it. When we are nowadays in the adulteration also, we see that not only the PD, for example, PD5 inhibitors, not only sildenafil, tadalafil, vardenafil, but their analogs are being used. And the imperceptible use of these analogs is very dangerous because they have not been tested formally for the safety and efficacy. Hence, in view of the potential harm to the public health, effective and proactive measures are required to guard against the illicit use of these pharmaceutical analogs. So basically, there are in each class. Sometimes it is locked. I don't know. So there are in analgesic drugs. They are using this paracetamol, diclofenac. In the case of cybertamine hydrochloride is used in slimming drugs. Similarly, PD5 inhibitors are used in the sex stimulant. So these are the difficulties which activists, or you can say researchers, have to find out for the safety and efficacy, the safety of the herbal drugs. And that is why I said that these instruments are important. And these genomics and metabolomics, this seems to be the future, irrespective of the fact that they are very costly. With this, I will once again thank to the organizers for giving me a chance to be here. I am also thankful to our Chairperson, Madam Amina Al Haidan, for encouraging me to present this finding. And last but not of least importance, I'm thankful to all the audience for listening through and through with concentration. Thank you very much.